My name is Vitor Pereira, and I would like to thank Saravana and the team in Singapore for the invitation. I apologize for yesterday's lecture. I've been coming from a conference in Argentina, and I missed a couple of connections. It's been hard to be back here in Toronto. So I'm very happy to share with you our experience with computational fluid dynamics in neurovascular diseases. I know that is sometimes a basic science topic, but it's something that we've been working on it in many years, and it's quite passionate. So computational fluid dynamics has been used in the industry for a long time. So some industries like car industry, some science projects are completely designed and planned using computational fluid dynamics. The modern planes, they are all designed until their final stages of prototyping with virtual simulation and computational fluid dynamics. So this has a huge potential and it's already being applied in different industries. When we speak about the body, and particularly for us in neurointervention, the computational fluid dynamics has the power to study and simulate the hemodynamics, which is the blood circulation and function of the cardiac cycle. And it encompasses the whole cardiac system, the arteries, the heart, the vessels, and the diseases that can affect these systems, including atherosclerosis, aneurysms, and I will introduce to you venous disorders that we also have been applying computational fluid dynamics. And it can give us information on the natural history or impact on the flow in the natural history, and also can support and help the development of new technologies. So the blood is an important part of it. So the composition of the blood plays some role in the hemodynamics. The arterial dynamics plays also another play a role as well in this equation. And it's important that the flow may affect the blood may affect the flow and the vessels. So it's a system that cannot be separated and isolated from the others. And there are many fundamental laws that regulate or that rule over the hemodynamics and the planning and the computational fluid dynamics of it. But the main one is the equation of Navier-Stokes, which actually is the equation that allows the simulation of the flow itself. We have to assume a number of parameters that will be invariable, so the equations are not that complex, but this has been the basis of the study of the flow in different materials, and that is the basis of the application of computational fluid dynamics in neurovascular diseases. And when we see the blood flow, we actually try to determine two types of flow. One is the laminar flow, which is the flow that runs in layers in parallel, which is the flow in the majority of middle and small vessels. And we also have the turbulent flow, which is a chaotic flow that is actually present in the aorta, the big vessels in the heart, and also in some narrowings, some aneurysms, which is also one of the reasons that, or one of the targets of computational fluid dynamics is to determine these patterns, correlate them with specific diseases, and actually gather the information that we can get from computational fluid dynamics and apply on the prediction of the natural history or on the planification of treatment of these diseases. So in medicine, computational fluid dynamics is applied in many different domains, in the blood, in the bone, in the lungs, in the cardiac cycles. There are methods that use MRI flow to get patient-specific parameters to apply CFD. So it's well known in cardiology, and there's already some clinical tools on it. And this is a field that is constantly evolving. in neuroscience, we will talk about actually two systems. And the way we derive the computational fluid dynamic is basically one single method that we can actually get from the imaging to the computational result at the end, and is where all the images in medicine or all the applications in medicine are derived. So it starts with a process that we call segmentation, in which we take the imaging acquisition in 3 dimensions for the vessels, and we filter, we cut, we smooth, and we create something that we call geometrical or 3-dimensional geometry. So once we remove the bone, we remove the parenchyma, and we smooth, and we have the good anatomy of the vessels, we produce something that we call meshing, so which is that 3-dimensional view, but we gave the spatial information to the computational system that will actually apply the Navier-Stokes equations with the parameters that we will determine or give to the computer, and then after a few hours or days, depending on how elaborate or how complex you want your simulation, you can get the hemodynamic information. So there are different softwares that can produce the segmentation and some different methods, and there are also different simulation systems and products that will actually be used to calculate these equations. So depending on which variables and how precise you want to study, you need certain software. So we use something called Phoenix, which is a solver that is used in mainly the engineering universities, and is one of the projects that the Canadian engineering universities have. And since we use high definition computational fluid dynamics, this is the solver that we actually use. And this is the process that I was talking about, the meshing, which takes the 3D and actually subdivide this 3D in very small pieces, and that's what is sent to the solver. And the flow is calculated in all these pieces and then fused together, and then we can have specific parameters for every different region. And this is a case that was actually done almost 15 years ago. It's a case from a master thesis, a PCOM aneurysm that was treated and recanalized. That was the initial aim of studying the flow in aneurysms, at least on in my master thesis at that time. And we actually used computational fluid dynamics to look for hemodynamic parameters that could predict the recanalization of coiled aneurysms with or without balloons. And in the far left, we have the 3-dimension, and in the center, we have the flow and the different streamlines, those small tubes showing the direction of the flow. We call them streamlines. And on the surface of the artery, these colors indicate a variable which is called the Walsh stress, which is the interaction between the blood flow and the arterial wall. We assume for this kind of simulation that the arterial wall is static, so there's no pulsatility, so it's a solid vessel, which is, I know, a limitation, but otherwise it would be almost impossible, very complex, to produce a simulation in a vessel that pulsates. So with that, we can calculate the Walsh stress. And in those specific zones, you can see here at the level of the neck, there is a high Walsh stress zone that at the time that we did this work, we correlated these Walsh stress zones with points that actually correlated with recanalization. So translating to today, we could assume that if we had this pattern in an aneurysm that we would treat with only coils, this is a case that would require an adjunctive device a stent or could be even an intrasaccular case uh, to prevent that high wall shear stress zone that is in red at the level of the neck of producing a recurrence. So we will talk about today on the computational fluid dynamics in two diseases, in brain aneurysms and intracranial venous diseases, particularly on intracranial hypertension and pulsatile tinnitus. So for brain aneurysms, as different other areas in medicine, we are looking for biomarkers. We know that there are different types of aneurysms, and particularly the saccular ones. We need to know, once we diagnose an ruptured case, what is the natural history, if it will grow, if it will remain stable, and if it will rupture. So this has been a long-term research question that has been unanswered. So today we still use size and the other risk factors, cardiovascular risk factors, to define the treatment of an aneurysm and the natural history. Nothing else, no gene expression, no computational fluid dynamics, no imaging method can determine and help us differentiate. So we have been using computational fluid dynamics to try to answer this question. And let's see what is the current status. And the background is the interaction between the blood flow and the vessels. The wall shear stress will influence the endothelium and it will induce a response on the artery. So the blood flow in the arteries that are relevant for brain aneurysms, the mid-size arteries, it's a flow that has a very high velocity in the center and has a low velocity on the sides. It's a parallel flow, but it followed this pattern all along the vascular system in the brain. And depending on the wall shear stress, which is the interaction of the blood flow with the wall. So this is the variable that reproduces that interaction. It's not a collision, it's mainly the interaction because it has the speed and also the wall influence. So the wall shear stress can influence the endothelium. This is a work done by Adel Malek many years ago, in which he had this cell culture and he exposed those cells to different wall shear stress. And you can see here that the pattern, the distribution of these cells were dependent on the type of the wall shear stress. So the wall shear stress can influence the endothelial response. And with that background, that we started looking into flow patterns that could indicate a certain natural history on disease or a vascular disease. And we can see very subjective parameters like the jet flows. This is a paper from Juan Cerebro, a physicist that does a lot of research on CFD as well. And there are different ways that the blood flow enters and circulates inside the aneurysms, from simple to complex, or more specific parameters like the wall shear stress. There are different pressures and different parameters that have been produced or created to evaluate the impact on the flow and the wall. So this is just the illustration of some, and this is the list of some of these parameters: wall shear stress, oscillatory shear stress, energy loss, pressures loss, coefficient. So it doesn't matter what they name, so we can calculate them automatically once we have the CFD. It's just that for some aneurysms, some will be more or less relevant than the others. And this is one of the first papers indicating that the low wall shear stress is actually independent associated with the rupture of certain types of aneurysms. They did a study here in patients with MCA aneurysms, and they were independently associated. So this is a table of one of our papers retrospectively reviewing almost 200 cases, and we could not find the same power of the wall shear stress. Actually, the aspect ratio was more relevant than the wall shear stress. So, and what is the difference? So one study shows something, another study shows something else. So we started observing along the years that differences in techniques would produce different results, and some parameters will be more relevant to a certain location or using a certain technique. And the geometry and the rupture status also influence these results. So if we got the 3D geometry after the rupture, these aneurysms will likely to have the rupture point as a low Washer stress zone, and it will produce an artifact on the results. And then this would confound and influence the results of the CFD. This was one of the reasons that different centers had actually different results. So the challenge here is then finding prospective imaging cases that will actually will show the aneurysm before the rupture without the geometrical change that the rupture would produce. And this is still a challenge that we have to face until today. This is an Australian fellow that produced this paper. He had pre-rupture geometry and a post-rupture geometry. And you can see here that a whole lobule completely disappear, and it does influence the outcome of the computational fluid dynamics. So this is an artifact. This is something that influenced the validity. And since the computational fluid dynamic for aneurysms looking to precision, looking to a very well acquired imaging, so these results started to be hard to make sense and to reproduce. And different studies again would show different results. Some variables would show relevance to different locations. And this is an interesting paper. So we actually had 4 cases that we had pre-rupture geometries, and we did a matched controls with other locations, ACOM, Basel, MCA, and PICA. And we actually looked into all the parameters. None were positive. And we actually saw that the distribution of the Walsh stress was different per location. And the cases were different than controls, except the Basel's in every different location. So we described that actually the Walsh stress is location dependent, and the results have to be analyzed and take in account the length of the imaging that you have plus the location of these aneurysms. We also reported a couple of years ago a summary of everything that had been published in computational fluid dynamics, and with these confusions in which variables we would take in account or not, we actually looked into everything. And for our surprise, the geometrical parameters, aneurysm size, aspect ratio, were more relevant and had a bigger power than the hemodynamic parameters. And this put a little bit of confusion, but it also demonstrated what we already suspected, that we needed to understand and probably change or standardize more the computational fluid dynamic methods if we wanted to study aneurysms. So the technical issues we described, this is a paper of a group from Germany that did a challenge. So a patient with 5 aneurysms and 1 had ruptured. So they sent these geometries, so the 3-dimensional picture, to different groups around the world, and they wanted to that every group would use computational fluid dynamics to predict the one that ruptured. So clinically, this PICA was the one that we saw from the beginning that this looks ugly, but this ACOM is also irregular. The MCA is the biggest one. So we didn't see the pattern of the subarachnoid hemorrhage. But just to show you the one that ruptured, which was the pica one, the differences in the geometry across the different groups that analyzed this case. So it's impossible. The geometry influenced the CFD results, and obviously only two groups could actually prove or demonstrate the difference between this aneurysm and the others, and could tell or could guess or suppose that this aneurysm was the ruptured one using computational fluid dynamics. This is another challenge on another aneurysm, and it illustrates the different ways that the CFD can be produced. So some people keep the vessel or the whole anatomy. Some people would just do the CFD of that small location. Some people create artificial vessels to simplify the calculations. So this all produce different results, and it does create confusion in the analysis of and the application of computational fluid dynamics. So the bottom line is that we need prospective data. We need to have the aneurysm before the rupture and have follow-up to understand which ones will be unstable, rupture, or grow in the future. And there are different studies now to look into computational fluid dynamics in a refined way using a high definition computational fluid dynamics, and this is what we've been working on. And we actually have been using a computational fluid dynamics that is 100 times more precise or defined than the regular computational fluid dynamics that we used to do in the past. And with that, we can see the turbulent flow. So in this video, you can see the jet flow inside the aneurysm, and these vibrations is called flow instabilities. We can actually measure because we actually see 1,000 to 10,000 time steps in one cardiac cycle. And with that, we can produce, or we can see the turbulent flow, and we can actually put that in account and evaluate if this chaotic flow can influence the behavior of the aneurysm. So there are different calculations. This is just one way that we use, and we've been validating and looking into different ways the natural history of the aneurysms, taking into account the turbulent flow. And one of the studies we are doing is actually looking into surgical pictures and looking into the turbulent flow and seeing the wall and trying to correlate the turbulence with the wall of the aneurysm. Obviously, that is a time aspect that, you know, we can control. For example, the small white area is an area of low wall shear stress, but a few years ago, it could be a thin area that actually had an inflammation and healed. So it's hard to determine the time, but we are trying to correlate. And also another study that we are doing is actually correlating the turbulence with enhancement on the wall. I know this is a controversial topic. Some centers or some studies demonstrate that there is a validity on prediction of aneurysm evolution, some not, but we've been trying to make sense of it. So this is every column here. There are different parameters, including the SPIs, which is the turbulent flow. The center row and the columns are each aneurysm, and the low row is the surgical pictures. And the second last row, this is the segmentation with painting. So we actually use us and the surgeons that actually did the surgery. We paint trying to reproduce on the 3D the images on the pictures that we saw in surgery. So we are working on progress. Again, the difficulty to analyze these results is to actually understand the time effect on the wall of a certain parameter and the influence on the inflammatory process. So the vessel wall imaging, we have been seeing a good correlation with the turbulent flow. We don't see correlation between the vessel wall imaging and the other flow parameters. So the wall shear stress doesn't correlate. Other parameters, oscillatory stress doesn't correlate. And the low row here, the SPI, we can actually see a good correlation with the cases that have actually enhanced on the wall shear stress. And to improve even our results, we have been measuring the flow using MRI or 4D MRI or 4D CTA. And we've seen that when we put the flow of the patient into the CFD, we can actually see better these turbulent flows. And sometimes when we put estimated flows that we use, calculate based on the diameter of the artery, we underestimate or we can't find optimal results with these very specific parameters. So big learning. So last year, after all these knowledge and this refinement of the CFD, I took that study that I've done 10 years ago, the matched case control series, and I applied these new and modern flows to evaluate the turbulence, and for my surprise was negative. It's showing that we still have a lot of work, particularly on the validation and understanding how these parameters will, or flow parameters or the flow patterns will influence the natural history of these aneurysms. So the neck is important. So we've been working on refining these methods, and we also have been working on actually listening to what the flow is inside the aneurysm to actually, like we do in surgery, but calculated before, see if there is any sound that will actually correspond to these turbulent flows. So this is being a study that brought us to our other field, which is actually the production of flow. And now we will talk about the flow into venous diseases. So the flow into aneurysms, we are still looking for the right methods and the good parameters to apply in clinical basis, but there is a lot of understanding that we got from the method and from the disease itself. And we started applying the CFD methods to actually the venous disease, the venous narrowings of the sinus producing pulsatile tinnitus or intracranial hypertension. And pulsatile tinnitus is a sound that is perceived but is not produced. So the hearing is a very important sense. It can change your mood, can change our lives. So like in this song, it immediately produces you something. And the tinnitus, imagine it's a sound that is repetitive, that has no source, that actually disturb the quality of life of the patients significantly. And the pulsatile tinnitus is actually a tinnitus, a noise that is pulsatile, following the cardiac beat. And this is one of the sounds of a patient that we were able to record, that they actually listen 24 hours a day. We pressed on the jugular, we were able to stop the flow. And this is, can you imagine, after listening a beautiful song, and how calming or influential in your mood it can be? Something like that will certainly impact the quality of life of the patients. And the pulsatile tinnitus can be produced by many diseases: arteriovenous fistulas, arterial diseases, skull-based tumors, and more recently, we've been seeing more and more the presence of venous diseases producing these sounds. And there are different venous diseases that can produce these sounds. The most common ones are the narrowing of the transverse sinus. And they can produce a turbulent flow that will be transmitted in different ways depending on the speed, the velocity of the flow, and how turbulent the flow will be, and the impact zone close to the inner ear. And the sounds can be transmitted as a vibration through the bone, or as a sound generated like a whooshing, a water running in a river through the air cells. And the patients will actually describe these sounds quite differently. And we started actually recording these sounds in these patients, and these are different spectrograms. So we take a high definition microphone, and this is just the sound, and you can see the patterns, and we can differentiate the different ways of propagation of this sound. And we've been applying the CFD process, which goes through the same method, segmentation, meshing, but we had to develop everything new for the veins, because the veins, we cannot get the same contrast to noise ratio. We had to produce meshing tools or algorithms that could extract the veins and smooth and send these geometries to a solver that then we had to calculate the flows and measure the flows with Doppler on the veins to put these parameters and actually study the flow, the velocities, and the production of turbulence to predict which cases may have a tinnitus that is caused by that specific narrowing. So these are different meshing tools that we've been developing here in our lab. And this is one of the cases that we have produced. Was a patient that we actually treated with pulsatile tinnitus shed and arachnoid granulation. Everybody was saying that this case should not be treated. We actually did the computational fluid dynamics, and this is the sound that actually the patient was hearing, and she told us that this is the specific sound I'm hearing. And this was produced by a turbulent flow just in the back of the temporal bone. And we have reported this method on a paper last year. And we actually observed that whenever these turbulent flows reach a certain threshold, the patients will describe a certain pattern of pulsatile tinnitus. This is important for us to understand which cases should be treated or not. We are at the beginning of this research, but it's just to bring it to you different aspects of computational dynamics and how we can use in our daily clinical practice. So to summarize, it's a powerful tool. We can evaluate the hemodynamics in vessels quite precisely. The hemodynamic aspect of the computational fluid dynamics is very precise. However, we are dealing with biology, we are dealing with the vessel, we are dealing with a biological interaction that depends on the epigenetic risk factors, on the genetic background of the patient. And we unfortunately don't have all this information for most of the vascular diseases. And also we see a lack of standardization. Different groups will do CFD differently. This is a process that is quite laborious. It takes still a lot of time, but I think whenever we identify one good parameter, one good biomarker, then it will be able to simplify these methods to actually get that specific parameter without getting all the data that we actually can get with CFD, but in a more straightforward way. So there's a lot of potential. We are also doing a prospective collection of data. So this is one of the studies that we are running, trying to pull all this hemodynamic and biological information to actually understand better the computational fluid dynamics results. And we think that this is a powerful tool for now on the research level, but has a lot of potential to be used clinically in the future. With that, I thank you for your invitation, for your attention. I hope the conference has been good, and I hope in the future I can be there in person with you. I have to share the credits of this talk with my research group, my clinical colleagues here, and particularly Dave Destaiman and Nicole Cancelleri, that actually were the other pillar on the imaging and on the engineering side of this research. Thank you very much.